Hi, my name is Cassandra Prober, and I'm a junior in the Hazen Plant Regulatory Genomics Lab at UMass Amherst, and I will be characterizing the effects of COBRA loss of function mutation in Brachypodium distachyon. To start off, I wanted to briefly review a little bit about plant cell anatomy. Every plant cell has a primary cell wall comprised of cellulose, pectic polysaccharides, and xyloglucans. After each new cell divides, it begins to form its primary wall first as it elongates. The primary wall is less rigid as to allow for changes in turgor pressure. After elongation ceases, cells may form a secondary wall that thickens inward. The secondary walls are comprised of cellulose, hemicelluloses, and lignins. The secondary cell wall is vital in providing the mechanical support necessary for the plant to stand upright, protect against pathogens, as well as facilitate water and nutrient transport via capillary action. As you can see in the right side of the screen, each of these dark lines represent the primary cell wall, and all of the pink that fills in inward is the secondary cell wall. Cellulose is a key biopolymer in both primary and secondary cell walls, and is in fact the most abundant biopolymer in the world. Cellulose is synthesized by the cellulose synthase complex. This transmembrane protein directly secretes chains of glucose into the cell wall. It is comprised of multiple ssA subunits and its associated proteins. The ssA complex aligns cellulose into organized microfibrils with crystalline structure. The higher the degree of crystallinity, the stiffer the walls due to increase in hydrogen bonding. One of the ssA proteins that has been previously studied in Arabidopsis is the COBRA protein. This is a glycosyl phosphatidylinositol anchored membrane protein and is speculated to anchor in the plasma membrane alongside the ssA complex to direct anisotropic cellular expansion in Arabidopsis, as seen in the image on the right. Loss of function of the COBRA protein in Arabidopsis resulted in reduced cellulose crystallinity and mispatterning of tissues throughout the plant. In order to better understand the function of COBRA, our lab studied its function in Brachypodium. Brachypodium is a model system for cereal crops. It's useful as a model because of its short lifespan, its simple growth requirements, and its small stature, which allows it to be easily grown in the lab. In addition, it has a small sequence relative to other grass models that could possibly be used, such as wheat. Using sodium azide, a point mutation was introduced, inserting an early stop codon in the place of tryptophan in the COBRA domain. This resulted in altered gross morphology and changes in the stem. Specifically, it was seen to have a singular shoot and large stem diameter with reduced secondary cell wall deposition. On the right, you can see our gene map, and on the top is our wild type COBRA domain. On the bottom is the truncated COBRA domain that has its inserted stop codon at 215. This BD-COB mutant appears to follow a recessive Mendelian inheritance pattern. Out of the 10 brachy that germinated from the sodium azide experiment, 7 of the mutants seemed to resemble wild-type Brachypodium, while 3 had a more severe single-tailor phenotype. We believe that the heterozygotes were crossed in order to produce these 3 recessive genotype plants. On the left, you are able to see that single tiller, reduced leaf growth, and increased diameter, as opposed to on the right side of the image where you can see multiple tillers as resembling the wild type brachy. Here you can see vibratome sections of the 3rd elongated internode in wild type on the left and our putative mutant on the right. These sections were stained using fluoroglucinol and HCl to stain for lignin. In the lower right-hand corner, there appears to be less thickened secondary cell walls in this fiber as compared to the wild type on the left. There also appears to be some mispatterning of the vascular bundles in this top image for our putative mutant. And there is a clear difference in the darkness of the staining, which alludes to less lignin concentration in the putative mutant versus the wild type. This all just allows us to visualize possible mispatterning of tissues that may arise from disruptions in cellulose deposition in the secondary cell wall due to a COBRA loss of function. As for our next steps, we're currently in the process of designing new primers for PCR and gel electrophoresis to readily identify BD-COB mutants. In order to validate the mutant sequence, Sanger sequencing can be used to identify that early stop codon. In order to visualize better in our histological experiments, we want to start targeting cellulose, so that can use either chalcophore white or Congo red staining to visualize cellulose in the cell walls. In addition, I would like to use the uptograph method of crystalline cellulose content estimation. This process eliminates hemicellulose and lignins from those secondary cell walls, isolating the cellulose, and then you're able to estimate the glucose monomers by a colorimetric anthrone assay. Thank you.